Thank you all for your patience. Thank you for joining today's webinar. As said already, I noticed that some of you have a quite strong background already in radio chemistry and radiopharmaceutical chemistry, so bear with me. Try to learn from the immuno-oncology aspects of this talk. And for those who are unfamiliar with the concepts, then we will take it nice and slowly from really the basics of how to track immune cells using positron emission tomography or PET. Next slide, please. So the aim of this webinar is to give you some command on how to understand how radiolabeled tracers can be employed to investigate the biodistribution of immune cells in vivo using non-invasive imaging. Then to outline the principal strategies scientists use to radiolabel immune cells. You should have an idea on how the imaging data is used in medicine and in drug development to develop more efficient and safer immunotherapies for cancer. And although the main focus of this talk will be in oncological applications, the radiotracers that are presented here are not limited in their use to tumor infiltrating lymphocytes and cell transplants for immunotherapy. You can think a host of other applications where they can be used, most topical one of those being the COVID-19 imaging, also imaging of organ transplantation and the associated graft versus host disease, and then all kinds of immunological autoimmune diseases, such as MS, for example, have imageable components that can be addressed using the tracers that I'm going to present. Next slide, please. So just a quick overview of positron emission tomography. It uses imaging probes radiolabeled with positron emitting radionuclides, such as fluorine 18, carbon 11, gallium 68, copper 64, zirconium 89, and iodine 124, to name a few. These are isotopes of interest for immune imaging, maybe apart from carbon 11, it's not really used. All have the physical half-lives in the order of minutes to hours and maximally a few days. And obviously, when we think of the scope of immunological responses and the time frame those occur in the body, we would like to be in the longer range of half-life, physical half-lives, in order to trace the cells. The imaging resolution, or the resolution of the PET image, is dependent on the energy of the emitted positron. And now, Sudeep, if you click, we get the animation there. Upon emission, the positron will annihilate with an electron, giving rise to two 511 keV coincident gamma rays, which are detected outside of the body. You can click again. And again. And using an imaging setup like described here, with a ring of detectors arranged around the subject, those coincident gamma rays can be detected. And this really gives positron emission tomography superior sensitivity and resolution, apart from the positron energies, but sensitivity, because these coincidence events are not very predominant in nature. All right, next slide. So just to go over some of the basics of molecular imaging tracer development, with my background in the imaging of nanomaterials, one of the things that I have come critically aware of, and I think this applies to immune cells, is that the incorporation of the imaging labels should not compromise affinity to the target. So Sudeep, if you click, there's animation. So if we think that this is our vector, it's a bowling ball, and click again, please, we put a flower on top, which represents our label. So you can click again. But if we add too many of those flowers, who knows what is underneath there, because all of the essential features of that bowling ball are now masked. So we don't know what we're looking at, whether it's a vase or a bowling ball. And this same situation happens really in the body, that the imaging labels can mask the essential features of the compound that we're hoping to trace and resulting in aberrant in vivo behavior, and the fact that our labeled compound does not act as a tracer anymore. So therefore, the characterization of the labeled conjugates in terms of immunoreactivity, if they're antibodies, target affinity and selectivity really with all tracers, cell uptake for those that are supposed to internalize, pharmacokinetic and elimination profiles and routes, and especially in the context of immune cells, in terms of cytotoxicity. So we have to know that the cytotoxicity of these immune cells towards cancer cells is being preserved upon labeling, has to be critically assessed. All right, we can move on. So just to maybe give you a little bit of my philosophy in molecular imaging tracer development. So you're quite familiar probably that, you know, the world is our playground in this respect, and biology in particular. We can think of developing imaging tracers literally out of anything, from low molecular weight compounds, you know, active pharmaceuticals, neurotransmitters, enzyme substrates, metabolites, in ascending molecular weight to peptides, antibodies, DNA, RNA, imaging of transgenes, whole cells, which is going to be the topic of this talk, and even pathogens or particles such as nanomaterials. So if you can click again. So in my view, the realm of tracer development is really the chemical modification, the radiochemistry that we do in order to incorporate that imaging labels and the associated bioconjugation techniques. And then the assurance that we achieve still analogous behavior in vivo to the tracer parent, the compound that we actually want to trace. And just in the schematic, you can really think of this realm as this triage of the label and its properties, the radionuclide, the chelator, you know, the chosen conjugation chemistry, then the properties of the targeting vector, and then the interactions thereof with the biological target. So it's highly multidisciplinary, and it is something that can be tremendously rewarding to embark on. Next slide, please. So brief overview on cancer immunotherapy. So what happens in cancer is a phenomenon known as cancer immunoediting. So when cancer begins in the body with the malignant transformation of a population of cells, what happens is that T cells and NK cells, natural killer cells of our body that survey the functions of the body, would detect this and go after those cells, destroy them, and the cancer would be gone before it even manifests. But what happens is that cancer cells actually evade these mechanisms, the recognition of the immune surveillance from the body, and instead they promote the growth of other immune cell populations in the tumor, and resultantly the cytotoxic T cells and NK cells will leave the cancer to grow. So if you click now, what we want to achieve by immunotherapy is to reverse this. So if you think that phases 1 and 2 are what are normally occurring in the body, we want to bring the body of the cancer patient back to those stages from the stages of 3 and 4 here. Next slide. This is just a quick overview on the diversity of immunotherapeutic agents that are out there. And I will focus on whatever is there on the left. You can click, there will be a little box to highlight that. So adaptively transferred T cells, different cytokines fall into also this category, checkpoint inhibitor antibodies, and then different vaccines that are supposed to elicit the endogenous cytotoxic T cell and NK cell populations to detect those cancer cells again. Then different formulations go into this. How do we deliver these agents, and by which routes? So this is kind of like the realm of drug delivery in cancer immunotherapy. We can move on. One of the foremost challenges in immunotherapy is really that reversal of the immunoediting or the cancer immunity cycle and the normalization of those processes. So a lot of the tumors are considered to be immunologically cold, which means that the cytotoxic B and NK cells are not present there. But you have populations of immunosuppressive immune cells, for example, the regulatory T cells. And usually this means that the tumor will have poor prognosis and poor response to immunotherapy. And one of the foremost questions is that how to convert these cold tumors into hot tumors, where we would have the CD8 positive T cells, the cytotoxic T cell population, and the NK cells, then suppression of the immunosuppressive cell types, which would result in improved prognosis and killing of the tumor cells when immunotherapy is being administered. You can click again, there was a little animation. So how to make this transition from cold to hot. Next slide. And one of the foremost and most successful strategies to date has been the use of chimeric antigen receptor or CAR T cells. And they are made so that you acquire T cells from the patient's blood and you transgenically insert a chimeric antigen receptor raised to recognize certain tumor-associated antigens. And then you expand the CAR T cell population so that you have a vast body of cytotoxic tumor-recognizing T cells that are being then infused back into the patient. They locate to those cancer cells and they will destroy them. You can move on. Here is a close-up of that. So the primary players here are the transgenic CAR, chimeric antigen receptor on the T cell, and the tumor-associated antigen that it recognizes on the tumor cell. You can move on. So one of the foremost challenges for CAR T cell immunotherapy, especially in solid tumors, and by solid tumors we mean tumors that are of masses, not hematological, where the tumor cells are being circulating and found in the myeloid compartment, or is the tumor heterogeneity and the antigen escape. So there in figure 1, you can see that the tumor-associated antigens are not really being presented adequately to the CAR T cells in vivo. Then the trafficking and infiltration of CAR T cells might be impaired, and it's quite difficult to achieve that in solid tumors because of the tumor microenvironment, which is immunosuppressive and nutrient restricted, which exhausts or decreases the survival of the CAR T cells in vivo. And at the same time, the tumor microenvironment is also very dense, which is difficult for the CAR T cells to even penetrate into. Next slide, please. And another strategy that is quite widely employed in cancer immunotherapy is immune checkpoint inhibition, for which the 2018 Nobel Prize in Physiology or Medicine was awarded to James P. Allison and Tasuku Honjo. And what they discovered was that, in fact, this immune suppression by cancer cells is mediated by something known as immune checkpoints, here shown by PD-1, which is programmed F1, a protein on the T cell surface that binds to PD-L1, so the ligand of programmed F1 on the tumor cells, and that prevents the cytotoxic attack of the T cell on the tumor cell. So what Allison and Honjo discovered was that you can actually use neutralizing antibodies to prevent this PD-1, PD-L1 interaction from taking place. CTLA-4 is another widely described immune checkpoint inhibitor, which reactivates the ability of the T cell to destroy the cancer cell, resulting in cell death. And a lot of efforts in this area have been directed into research into this area. A lot of clinical trials are ongoing, but so far the results have been quite modest, and definitely not up to the promise in solid cancers. You can move on. So now the question is, what can PET do in immunotherapy to improve this? So in cancer immunotherapy, PET imaging can provide quantitative visualization on the tumor accumulation and persistence of adaptively transferred cell populations. We can visualize the reversal of the cancer immune evasion. And furthermore, you can look at the tumor infiltration and activation of endogenous cytotoxic lymphocytes, so CD8 positive T cells and NK cells. If you click. So this improves the patient selection for immunotherapy and helps us understanding the factors contributing to the success and safety of immunotherapy. And it results in the improvement of therapeutic outcomes through combination of immunotherapy with other therapies such as radiotherapy or chemotherapy. Okay, next slide. So the primary strategies to radiolabel immune cells for BET are labeling of the whole cells before transplant, the CAR T cells and CAR NK cells, imaging of transgene expression, and immunopet imaging of immune checkpoint inhibitor antibodies and other T cell markers such as the CD8. We can click. And the first one is referred to as direct labeling, and the second one and third ones as indirect labeling of the cells. Yeah, next slide. So whole cell radiolabeling is by no means a very novel technique. Different lipophilic radiometal complexes have been used for decades to label cells. And the way they work, like gallium oxidation here, is that upon internalization, so first the lipophilic complex freely diffuses through the cell membrane, and upon internalization, the radiometal dissociates from the complex and cannot diffuse back because it's charged. The most commonly used complexes are those of hydroxyquinoline or oxy, radiolabeled with indium-111 for SPECT imaging, gallium-68, and zirconium-89 for PET. You can move. And this is actually from Anu Airaksison's work. I really enjoy this image. This is a SPECT-CT image of a mouse bearing of albumin-expressing B16 melanoma tumor. And the trace, the hotspots show the infiltration of albumin-targeted T cells that have been labeled with indium oxine. And there, the white arrow is pointing to the tumor. So this B16 OVA OT1 model is actually a very widely used syngeneic mouse model of adaptive cell transfer. So there we artificially use of albumin expression in the melanoma cells as the T cell target for the T cell receptor or the tumor-associated antigen. I think it's a very cool image, and obviously you can appreciate here the distribution of the radiolabeled cells in the immune compartments of the mouse. Next slide. So then here's a fantastic illustration from a recent review from Frontiers in Psychology by Gilbert Fruberth and his co-workers. He's a giant in the field in Europe. So transgenes are foremost used in the indirect imaging of adaptively transferred cells. And there is a very good reason to this. So first you start by transfecting the cells in order to elicit the transgene expression. Those transgenes can give rise to transporters on the cell surface. Most widely known is the sodium iodine iodized importer, or NIS, that can be addressed by just injection of radiolabeled or radioactive iodine. Then the transgenes can elicit the expression of receptors on the cell surface, for example, the somatostatin receptor or PSMA, and those could then be imaged using the respective radioligands, so different octreotide analogs, and then the peptide mimetics for PSMA. Then there is something known as the contrast-generating transgenes. So, for example, the herpes simplex virus 1 thymidine kinase enzyme, which can be imaged with fluorine-18 labeled FLT or fluorothymidine, and also the different luciferases. Although those are reporters for optical imaging, they do fall in this contrast-generating category of transgenes. Next slide. So now when we look into direct cell labeling and then the indirect cell labeling methods, we can already see that the foremost caveat in direct radiolabeling methods is that the label is gradually lost from the cells and their progeny, either when the cell dies, whatever radiometal that had accumulated intracellular because of the auxin, and complex dissociation will obviously be released, and there is no way that direct label would be transferred to any offspring of that T cell if it was to divide in vivo. So gradually we lose the signal and are not able to trace the persistence of the cell populations anymore. However, in the transgene-based indirect labeling methods, they do overcome this issue of the loss in signal intensity over time, but there is the drawback of having to do more additional genetic manipulation of the cell, and the need for multiple radiotracer injections for longitudinal imaging of the effects and persistence of the cell populations. So nevertheless, even though the persistence of reported gene expression means that the signal will stay and will stay detectable for a long time, it's actually logistically quite complex to achieve transgene imaging. Next slide. So to finish with, I want to show one alternative strategy, which is immunopath imaging to both transgenics and the whole cell labeling. This is an example from my recent work. So here we used asparagine B, which is the bacterial siderophore, a chelator for zirconium-89 to modify an antibody. You can continue. Then the antibody can subsequently be radiolabeled with zirconium-89, which is a beta emitter with a very appropriate half-life, physical half-life, to the biological half-life of monoclonal antibodies at high yield within 1 hour at plus 37 degrees. If you can move on. With radiolabeled antibodies, the determination of the immunoreactivity is really key. So we have to know that they will, even after the radiolabeling modifications remain, will recognize the antigen at high efficiency in vivo. And one of the problems in this type of research is that the endogenous tumor-infiltrating lymphocytes and NK cells are an extremely low abundance target, and isolation of enough of these cells for conventional saturation binding assays or LINMO assays to determine the radioimmunoconjugate immunoreactivity would require a really large number of mice, or the use of a transgenic cell model artificially expressing the antigen. So the way to work around this, and I maybe want to present this just because it's, to me, a very convenient opening in the analysis of radiolabeled antibodies, is the bead-based immunoreactivity assay, utilizing polyhistidine tagged antigens on magnetic nickel NTA beads. If you can click. So what happens here is that instead of using cells that express the antigen, we use the his-tagged antigens on magnetic beads to determine the immunoreactive fraction. So essentially, the amount of radiolabeled antibody that is recognizing or binding to the antigen decorated beads. My very good colleague, Shai Sharma from the Lewis Lab has actually put out a quite nice description on how to do this. So please look that up if you're interested in using the method. I have found it to be extremely convenient, especially with these low abundance targets, such as those immune checkpoints that I have developed tracers for. Next slide. And here is maybe a more curious or exotic example from my own work. So I worked with an murine immune checkpoint inhibitor antibody that is actually on the NK cells. And NK cells are just something that is sort of like up and coming in cancer immunotherapy. Recently, the first CAR NK, so chimeric antigen receptor NK products have been put out. They are looking to become quite promising alternative to T cells also. One of the immune checkpoints on NK cells is TIGIT, which is the T cell immunoreceptor with Ig and ETM domains. And here, just in the B16 F10 mouse model, so an immunologically, unfortunately, quite cold melanoma model, we already could see quite nice tumor uptake of the zirconium-89 labeled anti-TIGIT antibody persisting over several days. All right. Next slide. So with that, I think I hope that I have been able to give you a concise overview on how cells are imaged or can be imaged in immunotherapy, why we would be interested in doing that, and also some radiolabeling strategies and imaging strategies on how to go about imaging cancer immunotherapy. Thank you.
Thank you, Mirkka, for this extremely fascinating presentation. Do we have any questions? So if I look on the chat box, so there is no any questions at the moment. But do you have any questions directly to Mirkka? You can activate your microphone and ask questions. So meanwhile, I'm waiting for you to. Yes. So you can write also your question on the chat box as well if you are too shy to activate your microphone. But I can start. So Mirkka, you gave several examples on how to trace these immunoreceptor antibodies. Antibodies have, of course, this kind of drawback that they have really long circulation time, and therefore you need to use quite long-living radioisotopes for tracing them, and this way causing quite a lot of radiation exposure or long-lasting radiation exposure for the patient. So is there any other methods or approaches for addressing these immunoreceptors than the antibodies? So is it possible to make some engineered biomolecules targeting to those receptors?
This is a very interesting question. So it's a little bit of a holy grail, you know. I think the pharmaceutical companies would want to do away with antibodies at the first instance, because first of all, they are horribly expensive. So patient doses amount to, you know, tens of thousands of dollars. I know that there has been discovery into small molecule inhibitors of CTLA4, so the cytotoxic T lymphocyte antigen, which is one of the really widely researched immune checkpoint inhibitors. But the problem with all of this immune checkpoint inhibition is that they are actually protein-protein interactions. And now if you think about how difficult protein-protein interactions would be to drug selectively, I think it will be long waited before we see actually small or low molecular weight inhibitors that could address this problem. Personally, I'm thinking peptides. So I would say that we can reduce the molecular weight of the immuno construct, just going into smaller fragments of the antibody in order to improve the pharmacokinetics. Peptides identified through phage display, for example, would be quite interesting to see, but I haven't seen any reports that this is being done at the moment, which I think illustrates that this is a very, very topical and very difficult area to enter into. Then, of course, pre-targeted imaging methods. I don't know if those will be discussed here on the course, but methods where you actually use bioorthogonal reactions to trace antibodies, for example, that have a long biological half-life with radio tracers that do have a short physical half-life would be something that definitely warrants investigation into. But I think clinically, zirconium-89 labeling cannot really be surpassed at the moment for radiolabeling of antibodies, because it's the one where there is by far most clinical studies and safety assessment on. So I think we will be with indium and zirconium labeled antibodies for a long time in the future.
Thank you, Mirkka. So do we have any other questions at the moment from the audience?
I have one, but just a curiosity.
All right.
If I understood well, this kind of cancer therapy, immunotherapy, is mainly focused on the solid tumor. Is it correct?
No, actually, it works quite poorly in solid tumors. Mm-hmm. So the foremost uses have been in different lymphomas, so hematological cancers, and then in metastatic melanoma. And this is where the two FDA-approved CAR T-cell products are currently geared towards. The reason I cannot remember really from the top of my head why those would be so amenable to immunotherapy, but I think one of the things is that the T cells actually have a fighting chance of reaching those tumor cells in vivo, whereas in a lot of solid cancers, the tumor microenvironment is described to be hostile, so that it absolutely, because of the sheer physiological conditions, the density of the matrix, it is not a very livable environment for immune cells to be at, especially the cytotoxic T lymphocytes they actually need to physically migrate to the vicinity of the cell that they are going to destroy in order to release their cytotoxic effect. They cannot do it from a distance. So I think that's why solid tumors will remain a challenge. But there has been a lot of promise into, for example, the combination of tumor microenvironment modulating therapies, like hyaluronidases, in the context of enhancing adoptive cell transfer. So this is maybe something that will be emerging. Certainly, immunotherapy cannot address that alone, but maybe with the help of its friends, the traditional chemotherapies, I think it will come.
Thank you. And since I'm not, of course, an expert, and I don't have a a perception of the real situation in hospital. But this kind of cancer therapy is easily translated from in vitro, in vivo, and in clinical operative uses till now?
No, I think the foremost hurdle for the translation of preclinical findings is that mice and men are so different in this respect. So I have chosen to do most of my research on syngeneic immunocompetent mouse models, and that's unfortunately it's governed by research funding, of course, because if I was to move to humanized mouse models, so mice where the immune system has been sort of humanized, I could see the price tag of my research increasing like tenfold easily. I think one of the ways that we need to address this is that because imaging technologies are translational in itself, it doesn't matter if you, especially PET, it doesn't matter if you develop a good tracer for a mouse antibody in mice, because the same radiolabeling technology and the same imaging technology can be applied to human cells in humans, if you know what I mean. So it's we can do a lot of basic research by looking at mouse immunology, but then I think the power into clinical translation, working with those really expensive humanized models, and also patient-derived xenografts, which would be the most clinically relevant models, has to, eventually happen in order to warrant a clinical translation. But from the radiochemistry perspective, I think we're at a very happy place because of the translationality of the technique.
Do we have other questions? So I have one more question about the indium oxine, because this is really interesting radiopharmaceutical, because actually it has been in clinical use already for several decades. Mm-hmm. So it was originally developed for tracing white blood cells. And now during these new immunotherapies, it has experienced a new renaissance, I could say, because it makes it possible to label also the immunocells for these purposes. That was a really interesting, which you brought up, this gallium-68 labeled oxine. So what kind of, do you remember the details about the publication? So what kind of applications they have had? So how they have used the gallium-68 labeled oxine?
I think that some of the particular papers that I have read have used it to label red blood cells, but basically it should work the same. I think it has some kind of a stability drawback compared to indium oxine, but this certainly warrants investigation. I think into looking at zirconium oxine has had a few publications in the context of immune cell imaging. So I think everyone is kind of now trying to find what would work in this area. Certainly there are other complexes, tropolones, for example, that could be used. So I think it's something that we have to revisit and think, and then obviously as chemists we should think about the ways of how to make these tracers really to residualize and remain inside the cell. So this is something, there was just recently a very good paper about improving, I think it was in bioconjugate chemistry, about improving tumor accumulation by self-assembly of metal complexes. So if you macroaggregate sort of the radiometals intracellularly or in the tissue, I think this already then makes it more likely that they will not diffuse out.
Yeah, but then of course we always need to remember that the label dilutes, as you mentioned in your lecture, so that when the cell divides, so every time it divides the radioactivity also, and not always equally.
Yeah, exactly.
So it's not predictable in that way.